Okay, hi there. So in this video, I'll show you how you can visualize and analyze long reads with third-generation sequencing data. Let me first demonstrate the case with Illumina's paired-end short read data. Okay, let's open our samples. And in this study, we analyzed the line1 element in the first intron of the TTC28 gene. So, okay, you can see here that the whole genome sequencing data from this locus, and up here we have the TTC28 gene and its exons. Here we have a gap in the coverages, which is our line1 element, so let's zoom into that. Okay, so here you can see around 6,000 base pairs long line1 element, and the reads are in dark grey because their qualities are 0, so their alignment isn't unique. Okay, but let's look at this flanking region where we have lots of discordant reads. Okay, so if we click here, the pair for these reads, this one is in chromosome 4, this one is in chromosome 7, and in this one, okay, there seems to be some sort of small inversion, and if you double click here, you can see its mate here. If I have another example and I double click, this is opening a second window to chromosome 7, and if I double click this one, this is opening another window to chromosome 4, and you can spot the mates by just hovering your mouse over the reads. But I'm going to go ahead and just close these now because you get the idea, and as you can see, there is quite a mess at the flank of this line1 element. So let's go and now open Nanopore's third generation sequencing data. So we'll just open this. Okay, now in the bottom track appears our targeted data for this line element. You can see that the coverage is over 200. So now I'm going to zoom in again. Okay, so now these long reads are aligned uniquely also on top of the line element. And I'll click one of these reads for more information. So you can see the original fragment length is detailed here as over 5,000 base pairs, and here at the bottom of the box you can see that it's been split into two, and the length of this clicked read shows it's over 4,669. I can get more information about the other read again by hovering the mouse. And also by hovering within the box, I can see what reads correspond to what, like you can see me demonstrating here. Oh, and if I right click on the read, I can copy the sequence to the clipboard. Okay, so let's check out another one. We'll click here. And this one actually is a little bit more complex. This fragment is actually split into 4 distinct pieces or reads, and 2 of them have been aligned to different chromosomes. Okay, actually I don't need this track, so I'm just going to remove it quickly. But now if we double click this read, BasePlayer is going to split the screen between these target chromosomes. Okay, there are no other reads aligned to these locations, and their quality is 0, and this one is, well, close to 0. But okay, this is odd, so let's go out a little bit. Okay, this seems to be downstream of the IGF-1 gene, but okay, well, you can't really say anything with one read, so let's get another example. Okay, let's pick this one, for example. Yeah. Okay, now this is a little more interesting. This is a better example. So now we have lots of reads aligned also to the target site in chromosome 7 and with good quality. So, and again, we can see orientation of reads alongside the original fragment. Okay, so let's just check what gene this actually is in. And okay, so it's in the intron of this gene here. Okay, so let's close this now and see what we have. Well, now that we have these hundreds of reads at this locus, we'd really like to see all the places this line element has jumped. And there's a nice little Easter egg here. So if you type in third into the search bar and press enter, you can see a list of the target sites and how many reads support these. And for example, if I highlight this one, you can see there are 416 reads that are split to chromosome 7 and 14 in chromosome 4. And there are some random hits here and there. So yeah, okay, I guess we're done here, and you can find the link to the manuscript about this line element below, and see you next time.